So good morning, everyone. I'm Kristen. I'm founder and executive director of Longevity Global. We are a expert community of longevity researchers, entrepreneurs, and investors. And welcome to Community Pitch. And we'll just go ahead and get started with our first company.
Thank you very much, Kristen. My name is Robert Lustig. I am an emeritus professor of pediatrics in the Division of Endocrinology at the University of California, San Francisco. I'm also Chief Medical Officer of the startup Biolumen, and I'm going to try to explain to you what it is, why we do it, and how it relates to longevity. So first of all, the problem. As you know, longevity can't go anywhere until we fix the chronic metabolic disease pandemic, because all of the diseases that constitute type 2 diabetes, hypertension, dyslipidemia, cardiovascular disease, cancer, dementia, fatty liver disease, polycystic ovarian disease, these are all the diseases of mitochondria. And until we fix what's going on in the mitochondria, no attempts at improving or increasing lifespan are going to be effective. Now, the question is, what caused that mitochondrial dysfunction? And the answer is the transition of our ancestral diet to our current modern ultra-processed food diet. Our ancestral diet was a low sugar, high fiber diet. Now, the sugar, turns out, fructose is a direct mitochondrial toxin. It actually inhibits 3 enzymes necessary for normal mitochondrial function. But the fiber is also extraordinarily important, because the fiber is the food for the bacteria, and the fiber also inhibits the absorption of carbohydrate, in particular sugar, from the intestine into the bloodstream, thus protecting the liver. So the transition from our ancestral diet to our modern diet has led to this overwhelming global mitochondrial dysfunction and chronic metabolic disease to the tune of $10 trillion a year lost around the world. Now, numerous startups and numerous various venues have become big money in trying to deal with this problem, as you can see here. But none of them have actually solved the problem yet, and the reason is because they haven't solved the food problem. Fiber turns out to be extraordinarily important. If you take a look at this slide here, you can see that fiber consumption, it has a negative relationship to all-cause mortality, coronary disease, type 2 diabetes, colorectal cancer, and virtually every other chronic metabolic disease that we have. And there's a reason, because fiber is the food for the bacteria, and we are not feeding our bacteria. The combination of soluble and insoluble fiber does 6 things in your intestine, all of which are metabolically beneficial, all of which improve metabolic health. It impedes sugar absorption, reduces the insulin response, nourishes those intestinal microbiota, induces the satiety signal sooner, eradicates malignant cells from the colon, and the colonic bacteria produce short-chain fatty acids, which are anti-inflammatory and anti-Alzheimer's. Now, the food industry knows this, and they have been adding fiber to various foods. For instance, Fiber One bars. That fiber is soluble fiber, tends to be either pectin or inulin. And inulin is immunogenic on its own, turns out to not be such a good idea. Soluble fiber can do 2 of these 6 things. It can help the microbiota. It can synthesize short-chain fatty acids. Now, that's not bad, but it's not enough. Insoluble fiber would be like cardboard. If you swallowed cardboard, that would give you a satiety signal sooner and would eliminate cancer cells from the colon, but that doesn't do it either. The fact is that the 2 together are synergistic. The food industry cannot add insoluble fiber back because it's not miscible. You know, there's no way to put cardboard into a microwave lasagna. Well, that's what Biolumen is. Biolumen is an engineered but natural fiber. It's a set of fibers, actually. It is a microcellulose sponge, 7 microns across. When it is in its powder form, it is colorless, odorless, tasteless. It can be added to food. It can be baked into food. It can be added to ice cream. It can be added to bean dip. It can be added to yogurt. It can be added to anything that's not aqueous. As soon as it hits the stomach, these small microcellulose sponges start expanding. They end up expanding 70-fold. And when they do, just like the sponge at your kitchen countertop, the nooks and the crannies become available and open up. And inside, impregnated in those nooks and crannies, are a set of proprietary hydrogels, soluble fiber. Those hydrogels are all designed to be able to sequester and absorb glucose, fructose, sucrose, simple starches, so that they become unavailable for early absorption, thus reducing the glycemic response, reducing the insulinemic response, and protecting the liver. And because they don't get absorbed early, they go further down the intestine, where they ultimately feed the microbiota. They feed the gut. So 90% of the sugar in food is normally absorbed within the duodenum of the intestine in the first 30 minutes. The goal is to stop that. The goal is to prevent that from happening. And that's what Biolumen does. It sequesters, as soon as it hits the stomach, the fiber remains in the duodenum for about an hour, and then it traverses the small intestine over the course of the next 6 hours, where it feeds the bacteria. Sorry, there's a lot of noise. There is a in vitro model of the human intestine called the Scheim model in Ghent, Belgium, the company ProDigest. So this is basically like a human intestine ecosystem. It's Scheim stands for Simulated Human Intestinal Microbial Ecosystem. And in this model, we were able to show that Biolumen reduces the absorption of glucose by 33%, fructose by 36%, sucrose by 38%, reduces the absorption of starch by 9%, increases short-chain fatty acid production by 60%, which is enormous, and with virtually no side effects, only a 15% increase in carbon dioxide production over control. And that means essentially no GI discomfort. We have now undertaken clinical trials. We have done a 4-week double-blind placebo-controlled trial in Melbourne with the Royal Melbourne Institute of Technology. And the results, and I can show you those results in the question and answer period if you like, but basically we were able to reduce both systolic and diastolic blood pressure by 4 to 5 points. We were able to reduce percent body fat and also visceral fat by bioimpedance. And we were also able to enhance lean body or muscle mass, all of which are clearly beneficial metabolic parameters. So we think that Biolumen can basically turn apple juice back into apples in the intestine and allow processed food to continue to be sold, but have a metabolically healthy benefit rather than a metabolically detrimental one. So this will be an enormous boon for the food industry as they learn to retool to make metabolically healthy food over the course of the next 10 to 15 years. And we know from several studies that model the reduction in sugar in food that we can reduce the metabolic burden and also costs for fatty liver disease, heart disease, type 2 diabetes, and obesity. Biolumen is a painless way at the food industry level and also at the consumer level. Now, there are other people in this space, for instance, people who are working on gut health and also people who are working on weight loss. We think we will be effective in both and with no side effects and at a much lower price point. We've had a lot of traction since our first our offering last year. A 17-member team a double-blind placebo-controlled trial, and another one on its way. We have improved our formulation to a 7-micron material, which basically, as I said, is colorless, odorless, and tasteless. Can be added to chocolate, undetectable. It tastes better than the regular chocolate. And we are now geared up to be able to produce 1,000 kilos per month in our Shanghai facility. And we are opening for business on October 29th with a B2C internet. This is our team.
Sorry, you're at time. So if you could.
Well, that's good because this is the last slide. So thank you.
Yeah, go for it. Feel free to finish up. Yeah.
This is our team, and as you can see, our founder, Paolo Costa, myself, co-founder, and I'm happy to answer any questions from the audience.
Thanks so much for the presentation. We've got a couple questions in chat. Wari, do you want to ask your question out loud? I might be mispronouncing.
I see one here that says, what tools for measuring body composition in the Australia study? We only used bioimpedance in the Australia study. We are going to be doing a 6-month long-term mechanistic study at Rush Medical College starting early next year using the newest formulation. And this will be a double-blind placebo-controlled trial, and this will include DEXA scanning and other biomarkers of consequence. So right now, all we have is bioimpedance on body composition. Let's see. You will probably address this, but how is it consumed? Okay, so it can be consumed as a dietary supplement, as a pill, or it can be added to food. So the supplement would be a B2C type of arrangement, whereas adding it to food obviously would be a B2B. And we have several partners already that are lined up. The company Kuwaiti Danish Dairy Company in Kuwait, they are sort of the Nestle of the Middle East. They have committed to producing an energy bar that will have Biolumen as its basis. And I thank Delani for the kind compliment. Thank you. Any other questions?
Quick question. How does it, is there any noticeable side effects in the gut? Is that cause any GI disturbance?
So in the SHIME study, there was a 15% increase in carbon dioxide over control because obviously, there's a small amount of fermentation, but that's true with any fiber. I mean, in my world, it's fat or fart. So this is not at all surprising. In the Australia study, I will mention that anyone who had constipation who was on Biolumen, the constipation improved. Anyone who had diarrhea at baseline who went on Biolumen, the diarrhea improved. And we had no adverse events at all, and no one discontinued the Biolumen because of adverse events. So we had a 0 adverse event profile in 74 patients. So we are actually quite proud of our safety profile.
That's great. When can we try it?
You can try it now. We have samples.
Awesome. Great.
If you just tell us and we'll send them to you.
Perfect. Maybe we can have some for one of the upcoming Longevity Global events.
Absolutely. We'd be happy to.
Very good. Okay, any other questions? Feel free to unmute and jump in or put them in the chat. All right. Thank you very much.
Well, thank you. I appreciate your time. Thank you.
Have a great day. All right. Next we have Codi from Imaginostics.
Hello. Hi, I'm Codi. I'm here with Valerie, who's the COO of Imaginostics as well.
Nice to meet you.
Also my wonderful wife. Thank you for inviting me, Kristen. Can you hear me?
Yeah, we can hear you well. And
Cool.
Screen sharing should be enabled whenever you guys are ready.
All right, let me pull it up. put on my timer as well, so I know where I'm at. There we go. All right, so it's my pleasure to talk to you about Imaginostics. It's a company that I'm the scientific founder of. I'm an MRI physicist by training. I invented this technology that's the foundation of the company during my PhD, developed during my postdoc. After my postdoc at Mass General Hospital, I founded Imaginostics. And so it is a technology that's a platform technology in imaging. We are focused on Alzheimer's disease. And, you know, the problem in Alzheimer's disease is that really there's no way to detect it before symptoms. So it's this black box period of time between birth and symptoms where, you know, a lot of people are working on this. And of course, in terms of biomarkers of aging, there are no imaging biomarkers of aging. So I think that really differentiates us as well between blood-based biomarkers and other promising, awesome technologies that are coming up. And so currently MRI is really qualitative. So it's generating qualitative images that radiologists look at. If you're going to do something quantitative, you're going to have to do it at the group level. So then you're going to be looking at, for example, blood-brain barrier leakage. You're going to have 20 people that were hit in the head and 20 people that weren't. And then hopefully you can discriminate that signal. And so right now, MRI, there's like 40 million MRI scans a year, 20 million of those are with gadolinium. And those gadolinium images, again, aren't really quantitative, but that's how we visualize perfusion. And in terms of Alzheimer's disease, really the quantitative imaging biomarker has been for amyloid. Great, we have anti-amyloid drugs now. That's a huge, you know, amelioration to go from zero to some drugs for Alzheimer's. And so now we're looking beyond amyloid and we're looking for biomarkers that can target other parts of the biology of the disease. And so our solution is a 10x better MRI scan of the vasculature and quantitative imaging biomarkers. And so we're really interrogating vascular biology. And so for the first time, we can map vascular structure, function, and leakage. And so this is at the individual level. So that's really the differentiator as well over existing modalities. Our technology is compatible with iron supplements. So there's a ferromoxitol, which has been around for so long, it's now generic. So, and we're compatible with existing MRI scans and hardware. So really at the foundation of it, our solution is a physics-based, quantum physics-based technology where we aim to get a quantitative signal and enhance the vascular compartment with ferromoxitol for contrast, and then get quantitative vascular imaging biomarkers. And so there's many markets for this important platform technology. There's preventative diagnostics where you want to do early detection with specific clinics. I can tell you a little bit about that, where we're working now with a leader in preventative diagnostics. I can't tell you exactly the name of it because we're under a mutual NDA. And we're serving them now, going to do a clinical trial, 150 people. Those are their people who are within their organization, their clients. And services for drug development. We're now beginning commercialization to accelerate drug development. And so, again, you know, this is really important. The nice thing about this space is it doesn't require any FDA or regulatory approvals. It's really preclinical and clinical trials. And then really the goal, so, you know, the end goal for us is to be at every hospital. So get through any regulatory hurdles, end up at the hospital. The reason why the market is so huge is because, you know, we can do so much. So for example, why not replace all of gadolinium-based contrast agent imaging? 20 million scans a year in the US. Early detection of dementia is a really huge market as well. The Alzheimer's Association put out a facts and figures on how much money can be saved. You know, we're not haven't achieved it yet. But I mean, really, you know, looking at so many different diseases from cancer to other neurodegenerative diseases such as multiple sclerosis, it's really an important platform technology. The vasculature, the biology of the vasculature really describes so many different diseases. So happy to talk more about that. This is our team. So I'm the scientific founder. Valerie is a lawyer by training, our COO. Sandrine, you know, biostatistician out of the Broad. Chingwen was at Philips for 10 years, leading pulse sequence development there, joined us. Scott was faculty at Harvard Medical School, which is where I'm out of. He was there for 20 years, joined us. And Josh is just a really awesome engineer, does some image processing for us, currently a MD PhD candidate at Stanford. We hope to bring him on after he graduates in another couple of years. We're keeping him here. We love Josh. He's published a couple of important papers for us, including this work here. And just to give you an idea of the type of technology we do and what it is really in thinking about biomarkers of aging. And so here we're looking at APOE4, which is the single most significant genetic risk factor for Alzheimer's disease. And in Alzheimer's disease, we're well aware that you get neurodegeneration, you're going to get decrease in vascularity, and that's all happening later on. And so sort of the surprising thing that we found in this genetically modified knock-in model was that with very high statistical significance, Bonferroni correction, p-value divided by 174, which is the number of regions we're looking at, more than 50% of the brain is testing positive early on, the 20-year-old human equivalent, for being hypervascularized. And so the idea is that the brain is compensating for metabolic dysfunction by increasing the small vessel density. And so it's this idea that with these chronic diseases that we don't see until there's symptoms, these are developing in the brain throughout our lifetime and really should be detected. And so we go from these vascular images like that of the rat head to quantitative maps of the brain. And again, to give you an idea of sort of the motivation is, another study we published here where, we're able to measure blood-brain barrier leakage throughout the entire rat brain. In general, again, this is what happens in general on the left, sort of the competition. This is DCE MRI with gadolinium coming from a Brain 2020 publication. They got the control, the animals that were hit in the head. And the point is that to discriminate between these two groups is quite difficult. You need so many different animals. If you're a white dot or a yellow dot, individual measurements and individual animals don't really matter. We published a study with even more mild head impacts, less than 2.5% false positive leakage by total brain volume, and there's just no overlap between these groups. And so it's this idea of going from this sort of qualitative group-based statistical significance to something that's really quantitative and then useful at the individual level. And so we have developed this technology in humans. This is an example of 0.4 millimeter resolution in the brain, takes about 6 minutes to get. We did publish a paper where we compared the gadolinium-based contrast agent imaging in 20 people in pulmonary thoracic imaging. And there we did as good or better in terms of imaging the vessels at the resolution that was chosen in that study, which we also sort of dumbed down for the gadolinium imaging. But where we shined in that publication was radiologists judged us as being 10x better in soft tissue. So that's where we get the 10x better. Of course, you can see the quality of the images is outstanding. And then again, what we really care about is not those large blood vessels that we see that are kind of nice and useful for structural abnormalities, but quantifying everything in the small vessels. In those areas, you don't see the nice vessels popping out. Last year, we got some grants from the government. We got an award from the National Institute of Mental Health for mapping neurofunctional modulation in drugs. We got a grant from the National Institute on Aging for looking at diagnosing dementia early and mild cognitive impairment, and that was also backed by the Alzheimer's Drug Discovery Foundation. And so, yes, when we bring this technology to the leaders in the space who want to, accelerate important technologies, they've invested in us. And so now we have this clinical study now happening at NYU, Brigham Women Hospital, and University of Miami. We hope we'll make some money off this, but we are pre-revenue. We have our first clients that we're talking to for accelerating drug development, which is pretty cool. In terms of the technology, there's really nothing else that's quantitative and safe like ours for these type of measurements. We have 6 patents. There's many different partners that we have that we're either currently working with or applying for grants with. It's our funding and our strategic partners, and our software is available really on any MRI scanner. We are raising money. So, we have had this great investment from key opinion leaders. Now we'd like to also have some investment as well from outside who can help accelerate and development, pay for the team. We want to bring the whole team here to Lake Nona Medical City. Now it's Valerie, myself, and Chingwen. And so the other members are ready to come. So we do need additional outside investment. And with that, I thank you.
Thanks very much. Really enjoyed that. Do we have questions from the audience? Go.
Hey, I have a quick question. Very good presentation. Thank you so much. I wanted to just clarify, this is only a software. It doesn't have any MRI hardware in it, right?
That's right. It's a software. It's different than other MRI software companies in that we also have a pulse sequence that changes how the scanner operates and acquires data. And then we have image reconstruction that takes that raw data and makes images. And then we have another layer that takes those images, like the images you saw in that presentation, and computes biomarkers. So yes, it is only a software. Thank you for clarifying that too, because that's it. Sometimes people think we got a contrast agent to sell, and we don't.
Beautiful. Just a follow-up question. So the clinical work is on human at the moment, or it's only on mice?
No, that's right. So yeah, we're in the clinic. We just got our IRBs approved. It's at University of Miami and Brigham Women Hospital, where we're doing the MCI study. We're also doing developments at NYU. Previously, we did human studies at University of Wisconsin-Madison and UCSF as well.
Great.
A quick question here.
Yeah, go for it.
So since we are talking about longevity, have you compared your results on and see changes with age?
So that's a really important question. And so, yeah, I really like that idea. And that's so at NYU, we're developing and testing, but we're also doing our measurements in a younger population, so 18 to 35. And so our greater MCI study is focused on, people, the healthy controls in that study are 70 years old, right? So it is important to have sort of measurements at a healthy young age compared to healthy older age, and then compared to disease. So we will have those measurements from this study. And at the end of the day, when you have biomarkers for drug development, you got to have a target. And if you're a 25-year-old person and somebody tells you, hey, I can give you a drug that's going to give you the healthiest 70-year-old brain out there, it's not really attractive, right? So it's super important to characterize by aging, and we'll be doing that as part of this study, which is exciting. Yeah.
Okay.
Anybody else? Questions? All right. Well, I'm excited about this technology. I will get in touch with you guys offline. There's a couple of other companies that are synergistic with what you're doing. That would be cool to see if we can make some intros working on blood-brain barrier stuff. So that might be interested in your talk. It's very exciting.
Thank you.
Yeah. All right, everybody. So it looks like we do not actually have Gino joining us today. So this is going to be the end of today's community pitch. But I want to thank everybody for coming, and we really enjoyed the presentations. And feel free to get in touch with these folks offline. You can get in touch with them on Longevity Global Slack, or I'm sure they can share their contact information with you as well. Yeah, maybe if you guys are interested, you could pop your email in the chat. Our presenters, yeah, people can just take note, get in touch with you. Thanks very much. We really appreciate it. Have a great day.